All right, happy people are still here. The problem of having the second to last talk on the last day is you might be talking to an empty crowd, empty room. Anyways, so I'm one of BJ's grad students, and I've been trying to work at the interface of Markov state models and accelerated simulations for quite some time. And all this research started off with a kinase, where we were interested in simulating a DFG, and there are many kinases. This one is mine, it's called BTK. It undergoes large-scale conformational transitions, where this particular DFG out there sort of is going to flip around and then go into the DFG end state. And we were able to do these studies where we showed that the protonation had an effect on this kinase, similar to work that D.E. Shaw had done. And we were able to map out then complete free energy landscape, starting from just the crystal structures for this particular kinase. The problem was it took about 1.5 milliseconds of simulation. And so this was one of the papers I did in my PhD, and we wrote quite a few of those. And ultimately, you can run all these MD simulations you want, and you can build as many MSMs as you want, but 10, 20, 50 milliseconds is not even going to come close to the scale of the problem there is, where there are 500 kinases. I'm only going to show you a part of the kinome, and they all have at least one protonation that's important. They all have multiple phosphorylation sites, and there is hundreds of known mutations that have been predicted for each kinase. And there is multiple drugs that each kinase can sort of bind to. And what you want to be able to do is try to figure out how does the landscape shift as a function of all of these different things. And clearly there's a scaling problem, right? If everything requires a millisecond or hundreds of microseconds, this is never going to be useful for any real application until, I guess, quantum computers come along or something. And so the basic question that I sort of have been trying to answer for the last couple of years is, how do we scale mutation sampling by sharing data across simulations? And the fundamental, that comes down to, first of all, how do we accelerate molecular simulations from the start? And this is sort of being basically, how do you converge these simulations? And so there's like 40 years of enhanced sampling literature to choose from, right? Like there is, if you know relevant collective variables or orthogonal, or like reaction coordinates or some kind of these x-axes, what you're gonna call them from your system, what you can do is couple them to a high temperature bath of some kind and then accelerate your dynamics across them. And so this is something called metadynamics, where you're adding a Gaussian potential, which will sort of push you out of wells and push you into wells, and then that eventually will sort of equilibrate, and then the inverse of that gives you the free energy. In certain parts of Europe, this is actually called, it's not called metadynamics, it's actually called conformational flooding, and you can get yelled at if you use the wrong term. But you can also use something like Chris Chabot's adaptive biasing force. You can use temperature accelerated MD by Eric and Naden. You can use the evergreen like umbrella sampling scheme, but they all have this fundamental problem: is what is your collective variable? What is the reaction coordinate? How do you pick it for what I'm interested in mostly is a kinase? And it's not an easy problem. I mean, so in like one slide, the kinase has this important orange bit and important red bit. It could be maybe it's the distance of this residue to that residue. Maybe it's the distance of this residue to that residue. Maybe it's the angle that connects them. Or maybe it's something completely pathological. Maybe it's the square root of this distance multiplied by the angle plus the square root of another distance. And the problem is, in 10,000 dimensional spaces, our ability to intuit these is completely gone. And so how do you solve this problem? So what we tried to do was try to solve a simpler problem. What we said was, okay, let's say that we are able to run these simulations enough times that we're able to see some of these slow transitions once or twice. Can we then learn optimized representations of these coordinates so that the next time we run these simulations, we can do these kinds of, we can sample these modes many, many times. And the argument that we made is that the Tika coordinates, or the MSM eigenvectors themselves, are the collective variables that you need to use to accelerate these dynamics. They represent the slowest decorrelating degrees of freedom in your simulation. They control how quickly you get to equilibrium. And what you can do now is put them back into your simulation to enhance dynamics along them. And so as magical as Tika is, it's actually just a linear combination of some coefficients multiplied by some dihedral terms. And that works out of the box with something like Plumet. And so you can just copy the script and it will accelerate alanine for you, for example. And so initially the whole point here was that, okay, we can, let's say, like I have larger systems too, but let's say for smaller systems, even a day on the GPU for alanine might not even get you 1 or 2 transitions. But now you couple that with these kinds of MSM analyses, you learn the most optimal collective variables that you need to accelerate, and you can get many, many, many transitions. And so you can build an MSM on this particular curve right here, and you'll get something like the cyan region. Or you can actually just invert the free energies here and get the red curve. Now if you take this black curve and extend it out to 10 days, which would be somewhere over here, you will asymptotically reach the red free energies. But the whole point is to combine this with this to get the best of both worlds, where you now have the optimal collective variables from the Markov model, the free energies from the enhanced sampling, and you combine them in an optimal way to get the kinetics and thermodynamics, but now in a time scale that is much more amenable to simulations. And so the whole point is that if you're working at this interface, you can do a lot of other things. For example, if you have multiple coordinates in your MSM that you want to sample, you connect them via a biased exchange or Hamiltonian replica exchange scheme. Or maybe you can also use something which is known as the kernel trick for machine learning. And so for example, I said before, maybe your most optimal collective variable is the square root of a distance multiplied by an angle plus something else. That's just a nonlinear kernel or polynomial kernel for machine learning. You can put that into your Tekka model as a pre-featurization scheme, so it will learn all these non-optimal polynomial transformations. And then the Tekka will sort of find the combination you need to be able to accelerate the dynamics based on your current simulation data. And so we also extended this to neural networks, where we were like, okay, if we want nonlinearity, we should be able to take some simulation data and push it through some kind of nonlinear transformation, so why can't we just do a neural network, right? I mean, they can be used for pretty much everything nowadays. And so we came up with was an architecture that's similar to an autoencoder, but it's time lagged. So what you have is a protein coordinate, this is high dimensional. Over here, it gets compressed down to a single number. And then there's a variational layer, don't worry about that, and then it gets decompressed back to its future self. And so you can think of this as something like a poor propagator, and this as being an encoding. And that encoding becomes a collective variable that you can use in your molecular dynamics simulations to enhance the dynamics along the most relevant degrees of freedom. To be able to do this, we had to do some other tricks, and there's 4 people on the paper, and that's almost accepted at PRE, but actually before that got accepted, we actually were able to show that this can be a collective variable in this paper in JCTC. And yeah, so the whole idea here was, again, that you're basically running just enough time, you're doing this just enough work that you can run some dynamics, you can see some slow transitions, but then you can accelerate them many, many times. And you can make them transferable across different force fields, for example, so that you can, these nonlinear transformations can then converge the statistics for you much more quickly than having to wait for an MSM to converge. The thing is, it scales. For example, we can learn folding coordinates in these via these kinds of neural networks, and then you can actually put the entire network inside FlowMet. It's not the most efficient collective variable right now because it's a lot of work to compute these kinds of forces. But we actually now have a native implementation inside OpenMM that allows you to put these compute graphs onto a GPU, which would make it much more faster over time. And so the other thing, though, was that, okay, but what if this is too expensive, right? Like, who wants to do this amount of work for every single system they're interested in? Like, these 2 curves, like these 2 blips might be like a day for an alanine, but it might be like months and months and months for kinases. And so the question becomes, can we share data across simulations even further? And the whole idea was, yeah, you can. What you can do is now a sequence alignment and transfer the slow modes that you learned from one simulation to another simulation. Where, for example, if you're building some model on C alpha distances, for example, and you learn this transformation, which is a linear combination of the C alpha distances that might fold a protein, and now you make a mutation, all you need to do is map the C alpha indices to the new protein, and you transfer the weights over. And that allows you to, assuming that the collective variable, that the slowest transitions are transferable across these, and that the mutation doesn't induce anything new, you can transfer these optimal coordinates from one simulation to another. And again, the whole point here was we actually showed this in a paper that you can actually fold a GTT triple mutant based on the collective variable from the previous simulation that I showed. And are able to not only fold it in timescales that are, the total timescale I think right now is like 5 microseconds, but the individual trajectories are limited to 100 nanoseconds, and we have multiple walkers that sort of share everything. And so what we're trying to do is basically run one simulation learn these optimal coordinates, and then transfer the optimal coordinates to the next 100 simulations of mutations that you're interested in. Okay. So then there's still another problem here. Like, what if, and so this is like, this conference is interested in free energies. Like, what if you want to get around the entire MSM pipeline completely? What if you want to just build collective variables and not have to worry about it? Like, maybe what we want is just initial ideas about what the collective variables should be, and then move on. And the whole point is, let's say we don't want to run this simulation, or somebody else has never run this simulation, and so we don't want to do the work ourselves, so we don't want to learn from their simulation either. And I would argue that problem is something similar to, let's say you have a set of PDBs, and you're a grad student and you're told by a PI to go and try and fold this protein somehow. And what do? How do you build a collective variable that connects this particular set of PDBs, or maybe three different sets or something like that? And I think the problem is similar to if it was cats and dogs. But the idea is that what if you're told to learn a difference between what cats are and what dogs are? And what I argued in this paper was that these kinds of algorithms have already been developed by the supervised machine learning literature. What they do is they learn a hyperboundary that separates, for example, the folded state from the unfolded state, similar to how they would separate the cat state from the dog state. But then what you can do is use the distance to that boundary as your collective variable. It's a differentiable function that is high in the folded state and that's low in the unfolded state, and that allows you to move in between these boundaries in timescales now that are amenable to molecular simulations. But now we don't have to actually sample the transition using unbiased MD. All we need are the endpoints. And the point here is that there are a lot of different supervised ML algorithms that you can choose from, and I'm not so sure which one is the best one. But what you do is you start by simulating your end states for short timescales. Then you pick a very, very complete feature space. What you do is use dihedrals, distances, etc. Then what you do is you train a classifier that can distinguish between all of them. And there's a whole lot of different ones you can choose from, and you show that all of them sort of work pretty well. And then this function itself, the function that tells whether the protein is in state A or state B, is now the collective variable that will sort of push you between those regions. Okay, so coming back to the kinase, but since I only have 30 seconds, I'm going to go quickly. But you read all the papers you can to sort of build this dictionary of things you're interested in, which is like a lot of different residue sets. And then John Chodera comes on, and he's like, oh yeah, by the way, somebody already did this work for you. It's called CLIFS. Then we have all of our existing simulations, and we have all of our collective variable finding algorithms. Now what that allows us to do is build these transferable coordinates that will hopefully work for a large range of sequences. And what that allows you to do, for example, is transfer the DFG flipping coordinate from one sequence to another, and it goes through a different pathway. But more importantly, not only you can do this one time, you can do this many, many times across many different pathways. And like if you look at these trajectories, they all sort of are, like I tried to do simpler representations, but they all sort of go through different pathways, but they all sort of end up in similar DFG outstate regions. And this collective variable, again, we have never simulated Able before, but what I had was the BTK simulations, and what I had was this idea of what the residue set should be that corresponds, and then the mapping, the function that sort of transfers that into one number that tracks between the DFG in and DFG out is sort of what's put into these simulations. And okay, and so with that, I would actually like to thank my entire lab. They're pretty great. We take Halloween pretty seriously. Pretty much the entire code is on GitHub, so you can play around with it if you want. And we have a lot of great software that comes out of our lab, so feel free to use it. And then I use folding at home a lot. I use a lot of the GPU clusters a lot, so those are pretty great. And then I've been on Moneeb's NSF grants for a while. And thank you all for listening. Hi, John.
Super awesome work, Muneeb. It seems like the coup de grace would be starting with your transferable coordinates and then on the fly learning anything else that happens to be slow and coupled with them that might be novel for the system.
Yeah, so
what's your take on that?
Oh, so I have been wanting to use the spectral gap work by Pertouche for a while. It's not in a form that I can use right now. It's like it's a MATLAB script. I don't really know how to read it properly. I will. Yeah, there is definitely work there that can be easily done. Yeah.
Okay. When you use Tika as collective variables,
Yeah.
do we have to somehow make sure that you use a kind of a sparse Tika? Because
Yes.
it's easy to imagine, right? If you use a Tika, it will depend on all the coordinates. If you use this as a collective variable, if you even transfer it, it will try to push all the atoms, right?
Yes, that's
to
the
like
point.
some state. So what's the importance of sparsity? Like,
Yeah, so the sparsity, so Robin McGibbon for our lab developed the L1 regularized version of Tika that sort of selects the minimal subset needed. And you use the GMRQ to sort of say, okay, at this point, this is the sparsity that makes the entire thing kaput. And then you pick, that's the cutoff you use. It's not going to be perfect, but I think there's low-hanging fruit here to like do just enough sparsity to get it done, because the collective variable doesn't have to be perfect.
Yeah. But it also should not involve the wrong atom, right, in a sense. Like, if you include the wrong atom,
But-
right, mess it up, right? If there's only one non-transferable atom, just break it, right, in a sense.
I don't think so. We can talk later.
Okay, yeah, let's discuss.
Mhm.